So today I'm going to talk a little bit about the broad area of microfluidics and then sort of narrow it down into a more pertinent area of bioprinting or using additive manufacturing in order to make biological materials, so biofabrication as such. So some of the work that I'm going to talk about are done by these two students of mine. So Rana was a PhD student. She just graduated earlier this month and continuing on as a postdoctoral researcher. And then Celine is a master's student. She started as an undergraduate a student doing some summers and thesis projects in my lab, and then moved on to a master's position in my lab as well. So I'll start off with this slide. It's called "What is Microfluidics?" Towards 2006 or so, Nature came up with a special issue on this topic area, and that they define microfluidics as the science and technology of systems that process fluids at very, very small scales. And by small, what we mean is 10 power 9, minus 9 to 10 power minus 18 liters or so, right? Using channels which has dimensions of tens to hundreds of micrometers. So it's a very functional definition, very physical definition of what microfluidics is. But all microfluidics also share some phenomenological similarities, right? One of the things that we see often in microfluidics is this purely laminar flow. So at very low, what we call in mechanical engineering as a Reynolds number, it's a number to characterize fluid flow. Fluid tends to flow in layers. They don't tend to mix or anything of that sort. So an example is what you see here. You have 5 or 6 channels merging into a single channel, and what you see is that these different colored fluids, which you normally expect to mix with each other, don't necessarily mix. Now that's a problem if you want things to mix, such as in an assay, but you can exploit that in a clever way if you want to do other things. So for example, if I want to stain the cell differentially, so for example I want the right hand side of this to be green in fluorescence and then left hand side to be red in fluorescence, then I can do that by flowing red fluorescence dye and green fluorescence dye in specific sections and be looking at transport phenomena within cells, for example, which is difficult to do otherwise. The other sort of major phenomena that we see is that surface tension is very dominant in these scales. So for example, this is an oil and water emulsion. These emulsions used to be unstable in macro scale, but in the micro scale they are extremely stable. So you can take a chip that has these kinds of emulsions, millions of droplets, shake them around, nothing ever happens to these droplets. So they are actually like physical walls that we see in the macro scale, right? And with that you can create these kinds of foamy structures which are interesting in drug delivery and other kinds of applications. But more importantly, with microfluidics you can parallelize. So this is an example here of 6 incubators in which you can do cell culture under different conditions. And it's not restricted to 6, you can make it into 100, you can make it into 1,000, and that scalability afforded by the fabrication technologies that go into making these microfluidic devices is what makes them more powerful. So you can do hundreds or thousands of experiments all at the same time and get the information that you need at the shortest possible time, right? So these are some of the sort of common advantages or phenomena associated with microfluidics. And with that, it has been applied in a number of areas. These are sort of the 3 broad and main areas in which microfluidics has been applied. So one area or the genesis of microfluidics started with diagnostic devices. So early in 1990s or so, some researchers started miniaturizing capillary electrophoresis devices in order to perform assays which took several hours to do in minutes or so. So the idea being that you can basically miniaturize what used to take place in a big lab onto a chip and automate it so that things can happen faster and it can be happening in the field rather than in the lab. So this was so-called lab on a chip devices. The idea was to shrink a lab onto a chip and completely automate it. So this is an example of some of the work that we are doing in our lab, where we are able to now take a sample and segment it, in this case into 10,000 smaller volumes very, very quickly, and we can now perform, if we instrument it the right way, 10,000 different assays all in parallel, for instance, right? The other area where microfluidics has been applied is in terms of medical research. Here, what you use is the capability of microfluidic devices to be on the same scale as the biological thing that you're studying. It could be cells or it could be small organisms like this. So for instance, we are using microfluidic devices in order to immobilize animals such as Drosophila larvae or C. elegans, and then basically image their entire nervous system at a single neuron level in response to a particular stimuli. So these were not possible before, but with tools that operate in the same scale as these organisms themselves, you would be able to do something like that. Another example is in the area of biofabrication, where you're able to create to some extent the same three-dimensional organization of cells, extracellular matrices and other inorganic materials that occur in natural tissues, and then try to mimic how they would behave in response to an external stimulus. It could be drugs, it could be other factors and so on. And that sort of will be the main theme of the talk today. Another big area where microfluidics is applied is in the area of biomedical devices. So this is an example of an artificial placenta device that we've been developing for about 3-4 years or so, where the idea is that babies that are born preterm with respiratory distress syndrome could be supported by a placenta type device that could be attached to the umbilical cord once the baby is born and could operate using the baby's heart as a pump itself and exchange oxygen to the ambient environment. So this is to support lung function for a couple of weeks till the baby grows up and the lungs automatically develop, and then there's no need for external support. So these are examples of the devices that we are looking at, and here microfluidics helps because we need very low priming volume for these devices. The baby's blood volume is probably around 80 to 100 microliters or so, and so if your device for oxygenation consumes half that volume, then you would have to inject new blood, and that can lead to additional complications and so on. And what you want is a device that is very, very small, but which is capable of exchanging at least 30% of the capacity that a normal term baby's lungs would provide, right? And so we have been having some success in that area. So these in general is sort of the broad view of where microfluidics is applied and how it is useful. What I'll be focusing on in this talk is this particular area of biofabrication. So biofabrication is technologies that are being developed to assemble cells, different types of cells, with various combinations of extracellular matrices in a three-dimensional form, integrated with vascular networks, so that we can mimic at least partially some of the functionalities that a three-dimensional tissue operates in, right? So I'll start with a historical view of how this area developed. So sometimes it's called tissue engineering, other times it's called biofabrication, and so on, just to give you a perspective of what was done in different time frames, and then arrive at what we are doing in my lab very recently. So why biofabrication? There are sort of three broad areas why you want to do this kind of thing. One is in the area of drug discovery, where people develop what is called organs on a chip, and the idea being that if you can mimic some functions of tissue type activity in these kinds of chips, then you can make many of these chips, run experiments in parallel, and use that to test drugs, to test natural products, to validate toxicological effect of these products, and so on and so forth. Not in humans, not in animals, but rather in these chips themselves. Another area of application is as in vivo bioreactors. So for instance, if you want to produce insulin in the body, you can basically create a three-dimensional tissue-like construct which has just one type of cell coated with a vasculature, and then perfuse or attach or implant it into the body, and then that could be able to take nutrients from the blood and then release whatever is lacking in the body itself. And then finally, and this is far into the future, is that if you are able to assemble all the kinds of cells in the right order with the correct kind of extracellular matrices and inorganic materials that are present in the body, along with vascular network to perfuse this and remove waste and so on and so forth, then you can start thinking of truly artificial organs which can then be implanted in humans, if not in animals, and so on and so forth. So that's sort of the three main reasons why people fiddle around with biofabrication in general, right? So where it got started was with two-dimensional type tissue cultures. So skin was one of the first areas that this got started in. This is an example, an early example from 79 of growth of epithelial cells, along with some fibroblasts in it, and that has been successfully made into a product. You could now buy these things and then put it onto wounds for better healing purposes and so on and so forth. The main reason why it got started in these kinds of tissues is that you don't need to build in vascular networks in order to perfuse them. So they are two-dimensional inherently, or maybe two-dimensional with some certain thickness associated with it, but you don't need vasculature for that. The next area that was explored was cartilage tissues. This is work from early 1990s by Bob Langer at MIT, and the idea was in this case not just do cell culture, but then to have some kind of a construct or a scaffold in order to do this three-dimensional organization rather than a two-dimensional organization. And so what he did was to use at that time what was non-woven textile technology, the idea being that you can have polymers which are biodegradable, extrude those polymers in some fashion, you can weave them and organize them in a particular fashion, or you can do cutting and crimping, which are traditional textile engineering operations. And then what could be done was to use a bank of needles in order to sort of poke them and to create artificial porosities in this. And so this is an example of one such scaffold that was built, and then subsequently you can basically immerse it into cells in a cell culture medium, and then the cells will start growing on the scaffold, and over a period of time the scaffold will degrade on its own, and then you get the kinds of tissues that you want. So cartilage was a good way to start doing that, and he was quite successful in doing this, and he's one of the pioneers in this area. So towards the end of 1990, Vacanti and Langer wrote an article in Lancet describing this area of biofabrication or tissue engineering, and the idea being that you have various types of cells, and they identified the number of different kinds of cells that one could incorporate in this strategy for three-dimensional tissue culture, and then combining them with matrices and then putting them into a bioreactor. So what you see here is a bioreactor where you can have these cells embedded in extracellular matrices, typically in a bead form, although it doesn't have to be. It could be in the case of a scaffold that you saw previously or whatnot. And then you have a global circulation. So you have in this vessel a global input and a global output that can be used to perfuse whatever fluid that you want. So this mimics sort of the systemic circulation that we have in the body, and then there will be beads or small pockets of different cell types that may be present, or it could be a monoculture of cells embedded in extracellular matrices. So the advance that they made was to think of it in the context of sort of three-dimensional embedding in a suitable extracellular matrices that could then be washed over or perfused over with cells, right? So even then, one of the things that they identified was that generation of complex and vascularized tissue and organs was the main challenge at that point. So although the approaches discussed above will meet the needs of many tissue types, fabrication of large tissues and whole organs de novo remains a major challenge. So this was around 2000 or so, where people were seriously thinking about integrating. How do we go about integrating vascular networks into these kinds of three-dimensional constructs of extracellular matrices that we are trying to build, right? So the clinicians came up with a solution for this, and the idea is that, well, if it is difficult to make vascular networks and structures, why not start with something that already have it, right? So Decellularized organs are a fantastic way to go about doing this. So you can take a pig's organ and then completely decellularize it by washing with detergents and other things, and over time you can remove it and all you have is the network with ECM, and then you can reseed it with cells. So Attalla, Badilek, and Marchioni were some of the pioneers who started this work. It's a fantastic idea. So you already have a three-dimensional network in there, and you can seed cells with it. And in some cases, such as in vivo bioreactors, for example, this is a very feasible approach. The only issue with it is that if you want to move further to create complex networks, and you want to determine or you want to place different kinds of cells in specific locations adjacent to each other and so on and so forth, it is often difficult to do in this case. One or random arrangement of a few cell types is possible, and then you would have to let them evolve and create the kinds of structures that they would eventually create. Right? Simultaneously, there were other people who were proposing a more engineering type solution. So this was Vladimir Mironov, Boland, and Fogas, and the idea was to use this 3D printing technology that was emerging at that time, for about 2 decades or so, but becoming cheaper around that time, to basically print a three-dimensional structure. And the 3D printing technology, the basic idea behind that is that if you have something that is in 3 dimension, all you have to do is to section it into 2-dimensional slices, right? And then if you can pattern those 2-dimensional slices in sequence and build them up together, then you get truly complex 3-dimensional patterns, right? And so the idea behind that was that, well, now you can start with this flat substrate, and then you can use software to take a 3-dimensional organ, section up it into 2-dimensional slices, and then determine where each constituent material, it could be cells, it could be extracellular matrices, it could be inorganic materials, whatnot, go in software completely, and then you will have a bunch of inks, right? These could correspond to cell types, it could correspond to extracellular matrices or an organic material, and then you will print based on that pattern of where that material is located in those 2-dimensional slices, and then you will repeat for each of those 2-dimensional slices in order to get the 3-dimensional structure that you would want, right? So that was the basic idea, and this is sort of the visualization of that. The idea, they imagined that it would be a series of nozzles which you can perfuse, and then what you want to do is to build layer by layer. So this is the layer by layer building of a cylindrical tube in this particular case. What they had in mind was tissue spheroids. So you can build these tissue spheroids and then print them, and through that they can then connect over a period of time and then build the structure that you want, right? So out of it came a company. So this company was called Organovo. Fogas started this company around 2007 or so, and they came up with this bioprinter called Novogen bioprinter, and this printer starts by printing these tissue spheroids in whatever form, and these spheroids merge with each other, and in order to create hollow tubes they use agarose as a sacrificial material. So that was then dissolved away. You can wash away the agarose by slightly elevating the temperature, and then you can create tube-like structures or other structures. So Organovo now makes well plates where you can have these tissue type structures, and then you can perform drug testing and so on and so forth. There were also other attempts to do this. So one way was to use microfabrication technology, which is prevalent in the electronic industry, in order to create patterns in gel-like material. So this is by Abraham Stroke in University of Pennsylvania, where he created these channel-like structures using a process called photolithography, which uses light in order to define structures and patterns in a material called photoresist, so that you can create a mold, and then you can cast gels on top of the molds in order to create these 3-dimensional structures or 2-dimensional channel type structures, and then you can merge another flat layer to it in order to then create perfusion networks in a thick layer of gel, right? So this was mainly done in agarose, and the reason for that is because then agarose you can basically melt and reform and melt and reform, and that was used as a way to bond these things. So this is an example where you have a channel made out of agarose gel, and then you can bond it to another layer of agarose by just elevating this temperature to about 60 or 70 degrees or so. So this works fine, but then you don't necessarily want to expose cells to elevated temperatures and so on. So this was a nice proof of concept, but not necessarily a practical way for all the kinds of things that you want to do, right? Others also use 3D printing, and so in this particular case you have a print of alternate materials, or two materials, one which is a collagen layer and then another one which is a gelatin layer, and the idea being that you can then wash off the gelatin while the collagen forms the three-dimensional structure. So these are some examples of channels made in that particular fashion. Again, this was another manifestation of the layer by layer approach in fabrication. The only issue with this is that you have to wash off these things, and when the channel network becomes too complex or too small, washing off becomes much more difficult. Another example was 3D printing using a scaffold that is made out of sugar. So it's almost like making cotton candy. The idea behind this is that you can build a 3D printer which can create three-dimensional structures in sugar, and that can then be dipped into a bath containing cells and ECM, and then subsequently you can gelate it, and then you can dissolve the sugar away in order to create a perfusion network. So again, that's a good way to do it, but then once you want to pattern different types of cells in different locations, it becomes much more difficult to do that. So what people then started doing was to create some kind of an assembly, a complex assembly, using extrusion type methods. So this is an example of a nozzle that was created where you have one solution coming in this case this is calcium chloride, and then you have another solution forming a sheath, and that sheath would be made of alginate, and therefore the calcium chloride will then solidify or gelate the alginate in the inner core, and then when you extrude this into a bath of calcium chloride, then you can get tubes formed. This could be solid tubes, this could be hollow tubes, but you have some kind of a tube formation in which you can sequence the materials that you're sending through. Various versions of this has been tried, so you can put it into a bath and use those tubes and then assemble those tubes, such as what has been done here, where those tubes have been assembled in a zigzag fashion, and then you pour another type of gel with a different type of cell in it on top of it in order to create a multi-material network. You can also do this using microfluidics. So this is an example from University of Toronto, from Melissa Radisic's and Axel Gunther's group, where you can use valves, microfabricated valves in a microfluidic nozzle in order to sequence the arrival of different materials. And then as it emerges into a calcium bath, you can basically freeze that organization of materials and then basically roll it over onto a pin in order to get sheets of materials which you can then experiment with. Right? So in this context, what we wanted to do was to make this process relatively simple and low cost. So all of these processes that you see, especially these use 3D printers which are fairly expensive, anywhere between $50,000 to $200,000 or so, and they all create structures which go into a calcium bath to solidify and then subsequently are assembled. So what we wanted to do was to do something that, one, is very low cost so that any lab can adapt this method. It has to be fast as well. And then what we wanted was a structure wherein you create a three-dimensional network and it is completely has perfusion channels through, and there's one inlet and one outlet, right? So that's easy to connect to as well. So we started with this 3D printer. It looks rather fidgety this was in 2012 or so, but it was very cheap. It was about $600 or so. And then this was meant to do 3D printing of thermoplastic materials, so similar 3D printers that you see widely available. We then took off the nozzle and then introduced our own nozzle, which was an L-shaped channel with a needle that is coaxially embedded into this channel. And in this you can flow calcium chloride in the middle and then alginate on the outside, very similar to what other people were doing. And the 3D printer provides you the ability to take a three-dimensional model, section it into series of layers of 2 dimensions, and then create a path for this nozzle to go through. So it was very easy to program. You don't need to build the whole thing, but you can attach your nozzle onto it, and then you can print biological materials rather than thermoplastic materials. So typically what people tend to do is to do this in a bath so that you get a solid structure formed. And when you do it in a bath, you get this kind of a tube-like formation, which is what everybody else got. So one day what Rana decided to do was not to do it in a calcium bath, but then put it on directly onto a dry substrate, right? And so I was predicting failure, but rather what she got was something that looks like this, where because of the calcium flowing in the inside, the inner core got stronger, so it had certain stiffness associated with it, while outside it was still not cross-linked, and therefore it would, when it hits the dry substrate, it spreads over a little bit, right? And over a period of time, the calcium actually diffuses out and then makes this structure stronger and stronger. And so what she did was create not just one line. So as you draw a line, you get this hollow core surrounded by a region of gel which solidifies over a period of time. Now what she did was not just draw a simple line, but then she created a pattern, so a zigzag kind of pattern back and forth. And when she did this, what she found was if she placed these tubes appropriately close to each other, maybe about 500 to about a millimeter or so in distance from each other, you can in fact create a thin slab of material completely perfused or having perfusion channels throughout it, and you can connect an inlet to it and an outlet to it and then perfuse that entire three-dimensional volume, right? So this was sort of the innovation that she came up with, and then she went on to do various patterns with it. So this is basically a back and forth zigzag pattern, and this is the channels in the gel. So this is how the thickness of the gel looks like. That's your hollow tube, and you have a thin layer of gel on top and the bottom and some layers on the side. And you can perfuse this with, in this case, a nanoparticle solution so that it doesn't diffuse away into the bulk of the material, and you can see the traces of the channel itself. Not only can you do one layer, but you can come back and do the second layer in an orthogonal kind of fashion, and you see here in this particular case you have one layer of channels going up and down, another layer of channels going back and forth, and so on and so forth. So you can now create about a centimeter or maybe 2 centimeters thick channel layers with the entire three-dimensional volume of the gel completely perfused with nutrients that you supply. So she, being in engineering, she went on to characterize the design space that you can operate in order to get good structures. So for example, in the green area you can get parallel tubes, while if you go into the blue area you get these flow instabilities which causes these coiling effects and so on, which may not be intended, right? And then she also characterized the fact that with the speed with which you're printing, you can actually change the channel dimension. So we can go anywhere between 500 micrometers in channel size to a few millimeters or so in channel size based on just the speed that we are operating this channel in. And you can smoothly and continuously vary the channel dimension. She then seeded this with some HUX and looked at the culture of it, and what you see is that bulk gel of same dimension, the cells die off after a period of time, while in the hollow tubed gels you have the cells growing still after 3 days, and we've done experiments for 7 days as well. Right? She then wanted to become more complex in the type of arrangements that she's doing. So instead of doing just one coaxial layer, she wanted to expand the capabilities to introduce as many as possible. So she wanted to find out whether this is a scalable approach in order to do this. And because, so for that purpose, what she did was instead of having one L-shaped channel with a coaxial needle, now you just repeat that pattern. So here you have 3 channels, and these needles are increasingly smaller as you go from the beginning to the end. So what you will have is calcium chloride flowing into the right center of all of these flows at the very end, while you have different kinds of gel materials loaded with different kinds of cells that then flow into these regions. And because we are operating in a purely laminar flow regime, these layers of fluid that are flowing coaxial to one another do not mix with each other. So you can clearly have nice boundaries between these layers and segregate the kinds of materials that you want in these layers with this kind of an approach, and then solidify all of them simultaneously through diffusion processes, right? So this is the basic idea. So this is how the flow in these devices look like. So this is we've just colored it differently in order to visualize this. This is the outermost flow. In this case, this is alginate. Here you have a second kind of alginate. This is the inner flow, dyed with a different dye. And then in the center over here, you have calcium actually flowing. You don't see that, but then the calcium seeps out radially and then polymerizes this material. And so what you see here is this blue. This is supposed to be blue color, and so that forms a nice layer. This is after it being printed onto a dry substrate. You get this nice layered structure where the center is hollow. You have one type of material with possibly some cells loaded in it, and then you have another type of material with possibly a different kind of cell loaded in it, right? So you can create these hierarchical structures in this particular fashion with this, and not only can you do 2, but you can also do 3. So here you have the first gel layer, the second gel layer, and the third gel layer, and the more you go outside, the more it spreads out. So that's the idea behind this. But the approach here was that to show that not only can you do 3, but if you want 10, you should be able to go and get 10 layers formed in this process. This is printing with that, so you can see that there are some demarcations in the layer. So these are the hollow channels back and forth, and then you can see that there's a layer that is close to the channels, and then there is a bulk layer in the middle, and you can have different types of cells in that, and you can also create multi-level constructs in this particular fashion. So these are some examples where she has gone on to seed different types of cells. I'm not sure if you're able to see this properly, but the idea is that here you have fibroblasts on the outside, you have HUVECs on the in the inner core close to the walls of the channel, and then you can see that this is the top slice and the center slice and the bottom slice according to this figure over here, and you can see that this is cylindrical in structure and distribution, right? We've also used different materials. So this is an example where in this case you have a hollow core, and then you have material type 1, and material type 1 in this case is just alginate. Material type 2 is collagen plus alginate. We can change it to fibrin and alginate as well. We use alginate as a structural material to hold the hollow core up. Otherwise, if we use other materials, just pure collagen or fibrin, then they tend to collapse by themselves. So we use a small amount of alginate mixed with fibrin and collagen, not for structural reasons. But what we find is that we can then grow different cell types in these natural ECMs rather than synthetic ones. So these are some cultures that she had shown. So with this, what we have is an ability to do this very rapidly. So this printing, for example, will take anywhere between 10 to 15 minutes or so in order to create this kind of an organization of few centimeters thick material, which you can have a perfusion through by connecting an inlet and an outlet to catheter type channel devices, and perfuse that entire volume, right? And the idea here is that now you have the ability to not only have multiple ECMs, but you can also incorporate multiple cells in a coaxial type arrangement, right? So she didn't stop there. What she also wanted to do was to investigate a different type of additive manufacturing technique, and this one is called layer by layer lamination. So one of the disadvantages of the previous approach, an extrusion based approach, is that if you want to print with valuable materials, suppose you have primary cells for instance, or you have a very expensive ECM like Matrigel and so on, it's difficult to do that because there's some wastage inherently involved in the syringe and the tubes connection and so on and so forth. So she wanted to create a completely different method where you can use expensive materials and still create this kind of inherent vascular network type perfusion channels. And the idea was to use layers to cast materials in the form of layers with these grooves serving, or patterns of grooves serving as sort of vascular interconnections, and then to use adhesives in order to attach these layers onto each other so that you can then build a stack of materials on top of it, right? So that was the basic idea. And many of the sort of tissue adhesives and other things that we use, what ended up happening was that they will typically clog these microchannels, right? So you create a layer of adhesive, put another layer on top of it, it clogs these microchannels up, and so we wanted to come up with a new kind of adhesive which would not do that, right? And so, so the planar processing leads to material conservation, and you can get very high resolutions because you're using this photolithographic process. And she investigated these two materials, silicon carbide and nanosilica, as inorganic adhesive materials in order to bond layers of ECM to each other, right? So nanosilica has previously been shown to bond tissues. So if you take two tissues, for example, put some nanosilica between them and attach them, you get these nice bonds, and you can use it as a tissue adhesive. The problem with that, however, is that it has to be used at very high concentrations, typically about 50% by volume or weight. And that forms a thick film. It's good for adhesion per se, but it's not necessarily good for viability of cells in the adjacent layers. The mechanism of binding is just van der Waals interaction. So proteins have certain domains in them that then tend to adhere to polarized molecules and polarized surfaces like silica. So we wanted to investigate alternate materials, and silicon carbide has even better properties than silica, so that is one of the things that we started investigating. And the idea behind that was to create these molds. These molds can be created very precisely. The channel sizes can be down to, say, 100 nanometers up to a few millimeters, if not centimeters or so. And what you can then do is to cast gels on top of it with different ECM materials that you want, and then you can peel them off. Those would be the individual layers, and because you can define these grooves as and when you want, you can create branch networks, you can create all kinds of different structures as you desire for perfusion purposes. So this is an example of two gels that are bonded together with a microchannel feature in it, and this one is bonded with silicon carbide, this one there's no adhesive in it, and you can immediately see that as you try to perfuse this, there's a big leak that happens between these layers. Here there's no leak that happens. She also characterized sort of the viability or the cytocompatibility of these materials and found that silicon carbide wasn't as different. In fact, the concentrations that we were using of the silicon carbide was extremely low. It's about 0.25% or so as compared to 50% for silica in that interface layer itself. And so she found that silicon carbide, there wasn't significant difference from the control gels that she was culturing. And then what she could do was to create these stacks. So here you can see different layers which are about 0.5 millimeters in thickness, stacked up upon each other with hollow channels going through them. And you can create features down to about 150 micrometers or so, and you can create branch networks in this fashion as well. We still use nanosilica, but nanosilica was found to be very effective in bonding collagen. So all of these gels were collagen containing gels, and so in bonding gels with artificial tubing. So in this case this is silicone tubing. If you want to bond plastics with gels or tissues, then this is an effective way to do that. So the interconnects were bonded with nanosilica, while the individual layers were bonded with silicon carbide or so. She then cultured, I don't have the data with me here, but she then cultured cells in them and found that as compared to bulk gels, the cells that are embedded in these matrices grow very well, and you can have different cell types in different layers and therefore get the heterogeneity that you want for testing, for drug discovery and other applications. So in summary, I'd like just to say that we've been developing two kinds of approaches for biofabrication. One is a very low-cost coaxial type approach where you want cells in layers around a hollow core which you can use for perfusion. The other one is where you have valuable cells and you want a branch network. Then you have this adhesive that you can use in order to attach different layers of proteinaceous extracellular matrices to each other in order to create a three-dimensional structure. So that's the end of my talk, and I'll be happy to answer any questions.